Alrighty, hello everyone, and welcome to the Research Brown Bag for the School of Public Health. I am really pleased to introduce our speaker today. Dr. Patrick Bradshaw is one of our faculty in the Epidemiology division, and he will be talking about analytical challenges in nutritional epidemiology and the promise of Bayesian methods for addressing some of those challenges. Thank you so much, Patrick, for making time to talk with us.
Great, happy to be here. So shall I go ahead and just jump in?
Yep.
Awesome. So yeah, I'll sort of jump right in because I can have a lot to talk about today. And why don't we, by the way, just save questions for the end, and then I think it'll be a little bit easier to kind of get through the talk today. So nutritional epidemiology is one of those fields that I think draws a lot of criticism because it's so difficult to do it correctly, to do it well. And I think a lot of that stems from the observational nature of nutritional epidemiology. And even when we have things like cohort studies, which are sort of one of the sort of gold standards of study designs in epidemiology, there's still a number of limitations that creep up or that sort of present a lot of challenges in nutrition. And these, there was a recent paper in Advances in Nutrition that sort of outlined a lot of these and compared the cohort studies to the sort of gold standard of RCTs. And the sources of bias really were sort of focused on sources of confounding, which is residual or unmeasured confounding, which tends to be happening a lot in observational research, selection bias, especially due to differential dropout, and measurement error. And the measurement error in particular is, I think, a little bit even broader of a concept, because here it's sort of framed in terms of error in dietary assessments, but honestly, there's really a lot of errors associated with all sorts of nutritional status variables, including things like body composition, physical activity, even we're talking about energy expenditure and things like that. So of course, none of these are unique to nutritional epidemiology, but I think a lot of these things do have some unique aspects in nutritional epidemiology that are kind of interesting to kind of work through and to think about ways we can address them. There was recently a really interesting paper in current epidemiologic reports that really framed a lot of the biases that I just outlined as missing data problems. And really, when you think about it, a lot of our biases really are missing data problems. Confounding can be thought of as a problem of missing an entire variable in your analysis. Measurement error is missing the true measurement of the variable. Selection bias can be thought of as missing a relevant portion of your study population. And so I think that this is just a really interesting way of framing a lot of the ways that we think about biases in epidemiology. And one of the really interesting ways to think about this is through the context of how the Bayesian paradigm can actually be brought in to address some of these things. Because the Bayesian paradigm works with uncertainty and missing sort of constructs really, really well, latent variables. And so just to kind of review really quickly, I have two slides that'll sort of hopefully orient us to what the Bayesian paradigm is sort of trying to address. The first is to just talk about the frequentist paradigm. And the frequentist paradigm is the paradigm that we typically are taught in most of our curriculum, and it's sort of the dominant paradigm in statistical inference. And this paradigm seeks to find the value of the model parameters that are most consistent with the data generating process. So it works with the probability or the sort of density of the data given the parameters beta, and beta is like the parameters from the distribution. And so it seeks to find the parameters that are most consistent with the data generating process. I wouldn't say to contrast, but sort of to present something slightly complementary in my mind is the Bayesian paradigm. And this characterizes knowledge of the model parameters before the analysis. So you say, going into my analysis, I think I know something about these parameters, and then it updates this state of knowledge after you view the data. And this can make some folks uncomfortable, and I'll kind of get into this a little bit later, but going in, we can actually codify almost anything that we know about the parameters, including that we know nothing about these parameters. So the Bayesian paradigm really does sort of encompass knowing, a wide range of information about the model parameters. And so loosely speaking, we deal with this thing called the posterior distribution, which is the piece on the left-hand side of this equal sign. And the posterior distribution in the Bayesian paradigm says that given our initial state of knowledge, which I'm going to call the prior distribution. So this is some sort of distributional statement about the parameters of our analysis. We can update this after we see the data. And what's really interesting, oops, sorry. what's really interesting and I think useful about the Bayesian paradigm is that parameters are interpreted really broadly. And so observations where you might have missing data are really considered parameters that you sort of impose priors on in your analysis. And so parameters are really interpreted broadly in terms of any latent aspect of the data generating process. And so as I said before, this whole concept of invoking priors or specifying priors makes some folks hesitant and it feels a little unseemly. But really, sort of strictly speaking, priors tend to only overwhelm the data if the priors are very strong or the data is very weak. And in those situations, you sort of want to know, you want to be sort of a little bit more cautious about imposing the priors. But in general, the priors sort of complement the data. And if you sort of think about the way that you would do a meta-analysis, for example, think about constructing a prior as all of the studies that you would sort of summarize into a sort of summary measure of an association, and then your data is that extra study that you just add on to the end. So conceptually speaking, like that's the sort of Bayesian thought process is I'm going to look at the state of knowledge before, and then I'm going to add my data to that in sort of a meta-analytic spirit. And so one of the things that's really appealing here is that priors can actually be non-informative. And so when this happens, when you sort of assert that you really don't know anything about the parameters, you actually get back the frequentist estimates. So it sort of equates to a frequentist analysis. And so this basically just reflects that you know very little about these model parameters. And we often do this as a sensitivity analysis. So even when we might be inclined to use informative priors or to incorporate prior information or previous information into our analysis, we will often do another sort of complementary analysis or maybe as a baseline analysis that says, I'm going to assume that I actually am not going to impose any priors, and then what are the priors doing by comparison? So this sort of really does give you a sort of broad overarching kind of approach to doing data analysis. What's really interesting though is there have been some studies that have shown that even weakly informative priors, even if you can impose just a little bit of information, they can really yield improved inference. And this is something that I think is particularly useful in areas like nutritional epidemiology, where we want to be objective, but we also maybe want to incorporate some prior information, but not something that's necessarily going to completely overwhelm the analysis. One thing that I really want to emphasize in the Bayesian paradigm is that informative priors, however informative they are, should never be arbitrary. You always want to inform these either through the state of knowledge in the field or through validation studies or something like that. They never should be arbitrary. They should always be well informed. And in reality, it's really not difficult to specify reasonable priors. So for example, in nutritional epidemiology, we rarely believe that a dietary component is going to be protective on the relative scale below 0.5, or that it's going to be harmful above a relative risk of 0.2, or an odds ratio above 2.0, or above higher than that. So you can sort of come up with a prior that basically says, I think it's most likely that it's in between these values, but leaving some possibility open that it could be less than 0.5 or it could be above 2. But you're weighting those extremes relatively lower than the parts in the middle. And I think that that's really an important aspect of the Bayesian statistical paradigm. So that was my crash course in Bayesian statistics. For those of you who are taking 250C next semester, you'll get something a little bit more detailed. But now I'm going to sort of dive into some examples that I kind of want to highlight about my experience with Bayesian methods and sort of what brought me into this sort of paradigm, into this sort of area. And so some of you have probably heard me talk about this before. This was really my first foray into Bayesian methods. But selection bias is actually one of these really tricky problems. It's very difficult to deal with in a frequentist setting, but can be made a little bit easier in the Bayesian paradigm. And so in diet and nutritional epidemiology in general, missing data on longitudinal exposures is often a concern. So we think that maybe those who remain in the study might be less prone to the outcome. They may be healthier somehow than those who drop out. And so whenever we think that lifestyle factors are related to survival, this can kind of impose this selection probability, this differential selection probability, and this sort of missingness mechanism. And so in my work, I was looking at a study of breast cancer survivors, and they're often motivated to make lifestyle changes, but it's really tricky to sort of see how those are related to survival for the reason that I just talked about. And so we really have limited evidence to support recommendations for cancer survivors. I'm going to talk about this a little bit later towards the end of my talk in another application. But the question I wanted to address was, how is weight change after diagnosis related to survival after breast cancer? Previous research only included those with complete longitudinal follow-up on exposures. And so basically we only selected, so previous research up to that point where I had begun studying it, had really only included folks who stayed in the study long enough to be able to measure these weight change variables. So the data that I was using, which is the Long Island breast cancer study, approximately 33% of the participants failed to complete the follow-up questionnaire. We did have some baseline data from them, but about a third of the participants failed to complete that follow-up. So, we wanted to be able to leverage the data that we had to be able to address people who had gone missing in the follow-up in order to address the relationship with cancer survival. So what we did is we used this approach called a selection model. This is something that was well established in the statistics literature. And what it does is it models the probability of being observed or missing, along with the covariates with missing values and the outcome. So if you sort of forget that first piece, if you forget the model for being observed or missing, modeling the missing values and the outcome is actually in the spirit of multiple imputation. So it's something that's actually very familiar to a lot of folks. But when you add in the modeling, the distribution of being observed or missing, this then becomes a selection model. And you can actually model the probability of being observed or missing as a function of covariates that might predict it, including mortality itself, which is something that I think is a really key aspect here, and this becomes very difficult to do in a frequentist paradigm, but not too difficult to incorporate in the Bayesian framework. So the first thing we would do is model a logistic regression for the probability something is observed or missing. So this seems very intuitive, and in fact, it actually sort of appeals to our intuition. So we're modeling the probability that something is observed or missing, given the mortality time, as well as the missing covariates and the observed covariates. So the XM are the ones that are missing, and the XO are the ones that are fully observed, the ones with no missing values. The next piece is the piece we really want to know. We want to know a model for the time to death, the time to mortality. This is the outcome model that if we had complete data, this is the model we really want to have. And so this model is sort of being weighted by this first model of observed or missing, as well as incorporating the missing covariates, which are then going to be modeled with their own model, either linear or logistic regression, depending on if they're continuous or binary variables. And so we're actually modeling the missing covariates. So if you take those last two pieces, the probability of the event time and then the probability of the missing data, that kind of sort of loosely corresponds to what you would get from multiple imputation. But it's this being able to incorporate the probability of being observed or not that makes this able to handle informative dropout. So the frequentist approach, you can actually do this, what I just outlined in a frequentist setting, but it's really computationally intensive and can actually become, it's really kind of hard to even get standard errors out of these estimates. But the Bayesian paradigm offers a really straightforward alternative in a way that doesn't really involve a lot of extra sort of computational machinery above and beyond what you would do in a standard Bayesian analysis. So what we want to do is be able to characterize the model parameters, which are the alpha, the beta, and the gamma, which are the parameters from the missingness model, from the proportional hazards model, and then from the model for the missing variables. And we do this through Markov chain Monte Carlo sampling. So when I applied this sort of model structure to a Bayesian framework, these are the results. And so this is sort of a little synopsis of the results really quickly. It's more comprehensively studied presented in the paper referenced below. But just to kind of orient us to this plot, the complete case analysis. So this is if I just ignored the missing data, I just threw out all the observations that had anything missing. You see that in the black line with the little square, and the square is the point estimate. So the hazard ratio for weight loss of greater than 5% is a little bit below 5. You see this is relatively wide confidence interval. For weight maintenance, this was our reference category. For weight gain, you actually see a little bit of a protective effect there. Again, we have very wide credible intervals. And then for a more extreme weight gain, we actually have a deleterious effect that's associated with greater risk of death. The blue line with the circle denotes the missing at random assumption, which is where we modeled the outcome and the missing covariate. So this is sort of analogous to doing multiple imputation. And as you can see, we're using some of the data here. We're using more of the data because now we're taking the data that we already have something observed, and we're using that to basically impute values for weight change and other variables that were missing from our follow-up questionnaire. And as you can see, when you recover some of that information, when you're able to use the information that you have, you actually get more precise estimates. So even for the weight loss, for the weight, and for both of the weight gain categories, these credible intervals are tighter than if you had thrown out the data. And that's actually very intuitive. This is what you would think would happen, right? Because when you're using less data, you should have less precise inferences, and that's actually what we're seeing here. Of course, these are all crossing the null at this point, except for the extreme weight gain. But nevertheless, I think the precision in the effect estimates is really interesting. So the red markers are when we're assuming the data mechanism is not missing at random. And so this is one where we made a more comprehensive model, where we were then modeling the probability someone was observed or missing as a function of all of these other covariates. And this is actually where we sort of start to see some kind of interesting things. We still see this improved precision, but we also start to see a little bit of a movement in the weight gain category, but nothing really notable here. But something that's important to note is that now we sort of feel like we've tried at least to accommodate the fact that some folks might be missing for reasons related to their outcome. And this is one of the really important pieces here in this analysis, I think. Oops, sorry. So apologies. I'm having leaning on my keyboard somehow, and it's unpredictable. So anyway, so the sort of take-home message here is that the missing data models, we observe these pronounced or precise increases in risk of death at the highest weight gain group versus complete case analysis. So again, we're getting more precise estimates in that last category. So in the complete case analysis, if we had just thrown out anything with missing data, we have this really imprecise estimate that actually includes the null. But when we actually incorporate missing data methods, including the selection model, we see tighter credible intervals that actually exclude the null. So we're actually able to sort of say have a little bit more resolution, a little bit more precision with our inferences. And so again, this allows us to use all the available data. I would interpret the weight loss associations very cautiously. I think that there's still a lot of probably unmeasured confounders or disease severity that we weren't able to accommodate there. We did try with what we had. We had stage variables, and we had other tumor characteristics, and we feel like we did the best job we could at trying to accommodate things that might influence the weight loss association, but it still remains. It's actually something that in cancer epidemiology these days is still not fully understood, is like how weight loss affects cancer survival. I would say that that's probably not intentional weight loss. That's probably unintentional weight loss in that lowest category. So again, the take-home messages are that the models that included selection probability suggested a little bias. So we didn't really seem like we had a lot of sort of selection bias, at least given with how we modeled it. But those models for the missing data mechanism tend to be sensitive to the parameterization of those models of missingness. So there was a really nice paper by Amy Herring in Applied Statistics where she did something. I actually modeled this approach after what she did. And one of the things that she showed is that if you even misspecify that missing data model by omitting an interaction term, you can get very different answers. So all of your conclusions with regards to that, those models that include the missingness mechanism are all really dependent upon how well you can model that missingness mechanism. So I would just say that these are best treated as sensitivity analyses. So I think that the main sort of inference should be made on the sort of the blue model there that included the models for the missing covariates, again, analogous to multiple imputation, but being able to model the missingness mechanism is a really useful sensitivity analysis, I think, and can kind of help you give a little bit more support to your conclusions. One thing I really want to emphasize about this method in particular, but all missing data methods, is that there's no substitute for good data. And so the objective here should not be to use fancy methods to make up for bad data. It's that we can explore the influences of missing data within the confines of some of these methods, but we're never going to be able to replicate the actual analysis we would get from good data. So I don't intend this to be any sort of endorsement for not being especially fastidious with gathering quality data and complete data. So moving on to another sort of source of bias is that I would call this information bias. And I would probably have a longer discussion with folks if they wanted to know how I really think about this, really think about the structure of this bias. But there's this observation in obesity epidemiology called the obesity paradox. And I promise to get to something nutrition related, diet related soon, but I wanted to give one more example of this. And there's been a lot of studies like similar to the one that I showed, where folks have actually not looked at weight change longitudinally, but they've just looked at body mass index, which is a measure of adiposity that, again, is controversial, and I'm going to explain why in a second. Folks have looked at a lot of studies of BMI and mortality along many diseases, not just cancer, but cancer is one of the ones that has created some confusion. And so it's identified the nadir, or the sort of lowest level of risk that's either within the overweight range, or what's commonly considered overweight, or within the range that's commonly considered to be obesity, which is the highest range of body mass index. And this has created a lot of confusion. In fact, there's some papers that have circulated about 5 or 6 years ago, where people were struggling with, well, does this indicate that we should be, advising patients with chronic disease to gain weight if BMI is positively associated with mortality? Well, this has obviously created a great debate about this in the literature, and this has kind of gone back and forth. And so even public health practitioners have not really resolved what this means as well. So one of the problems with body mass index, and I think really one of the things that I think is the limiting factor in it, and I think a lot of obesity researchers would agree, is that body mass index is correlated with both lean and fat mass. And so this makes it obviously really difficult to disentangle, is it adiposity or is it low lean mass that's really driving some of these associations? And so researchers have really been emphasizing the need for more relevant measures of body composition. And so there's been a lot of debate a few years ago about the obesity paradox in cancer survival research in particular. And I think that the consensus, even among other obesity researchers not involved in cancer, is even that body mass index just doesn't really do a good job of characterizing risk of different diseases. And so folks have really started to try to move towards more body composition focused metrics. But body composition is really, really tricky because it's really hard to get body composition measures in population-based studies. One of the ways that folks are doing it is with CT scans. And so there could be CT scans sitting in the medical record. I've been involved in some work with some collaborators at Kaiser that's been looking at that. And that's been somewhat elucidating, but I think for a lot of folks who work in population-based research, it's really tricky and really difficult to get anything that's a reasonable measure of body composition besides body mass index. This is why it's so ubiquitous. It's because it's easy. It's not that it's really that great. And so one of the I was working with some collaborators on survival in head and neck cancer, and we sort of noted this obesity, or really this BMI paradox, as a lot of people are calling it these days. And so if you just do a standard model for body mass index and head and neck cancer mortality, again, this is just a proportional hazards regression model, and I fit it with splines for BMI. We noticed that the nadir, the lowest point of risk, occurs solidly within the range of the overweight category as defined by the WHO. And so again, this is sort of, we have higher risk associated with lower values, but not much risk associated with the upper values. So we kind of have this sort of like tapering off point there. And so this is confusing, and I think this has been observed in a lot of other head and neck cancer studies as well. And so one of the things that I sort of came to mind was that there was this paper back in 1997 that said, well, body mass index can actually be conceived of as if you sort of take the numerator of this, which is your body weight divided to BMI is your body weight divided by your squared height in meters. The numerator can actually be separated apart into the body weight due to adipose tissue and the body weight due to lean tissue. And so when you separate this out, you can kind of create two new variables. One is called the fat mass index, or FMI, and one is called the lean mass index, or LMI. And these are simply the body mass index times the proportion of body fat, or 1 minus the proportion of body fat, respectively. And so David Allison showed that in a sort of a thought experiment, a sort of like toy example, that the convex relationship with BMI and mortality can actually be explained by opposing relationships between these two aspects of body composition. And so again, we really can't measure these in most studies. We have these in NHANES, or we have these in certain studies, but in large population-based epidemiologic studies, it's really problematic to get measures of body composition. So one of the things that I sort of thought about was that we can actually treat this unknown variable, this percent body fat that we wish we had, as a prior in our studies. And so it's very similar to kind of like thinking about the missing data scenario. Now we have a variable, this percent body fat, that's totally missing. So it's not like it's observed on some people or not, it's totally missing. So we can actually incorporate external data. We can use this auxiliary data to inform what that distribution looks like. So the first piece is the proportional hazards model. Again, this is the model we wish we had. We want to be able to model the percent body, something that incorporates percent body fat and BMI and other covariates as well. But then we can actually form a prior on the thing we don't know. We don't know that PBF variable, the percent body fat. We can model this, we can specify this distribution according to external data using NHANES. And so the outcome model now looks like this. So this is just a proportional hazards model. Again, we have the fat mass index, which is body mass index, just to make it explicit, times percent body fat. The lean mass index is the body mass index times 1 minus the percent body fat. And we're adjusting for other variables like age, sex, race, smoking, some dietary factors. And then we specify this percent body fat, this missing percent body fat, using a prior, which is a function of body mass index plus some other variables that we think might explain percent body fat, such as age, race, sex, and smoking. So using this, I specified 3 models because in a Bayesian analysis, I think it's always reasonable or good practice to do some sensitivity analyses. I took the models that I fit from NHANES and I tweaked the parameters a little bit to basically alter what the information I'm sort of imposing on my final analysis is. So the first model is I just took the models right from NHANES, the parameters right from my NHANES model and just applied them to the approach that I just said before. But then I started playing around with the relationship between the BMI and percent body fat. So I attenuated that coefficient. So I basically tried to impose a more attenuated relationship between BMI and percent body fat. And then I also did another model where I took the residual error on that body fat model, and basically I made percent body fat more variable. So it would have a lot more variability in my final analysis. And what we found was actually really consistent with that the BMI mortality relationship we saw before was consistent with an increase in risk of death associated with greater fat mass index and a decrease in risk of death associated with lean mass index. So this is actually these are almost exactly the same parameters that David Allison did in his little sort of thought example before, but now we're able to actually apply this in an actual analysis and get credible intervals, which are the Bayesian versions of confidence intervals around them. So this was really one of the I think first times that Bayesian methods had sort of been used in this sort of an application and a sensitivity analysis for something like this. So I think, like, what is the utility of this? So I would not ever say that this is necessarily going to give us firm, definitive conclusions on the relationship between body composition and cancer mortality. But when we're faced with paradoxical relationships with something like BMI, which we know is not a great measure of adiposity, we can use this sort of an approach, the sensitivity analysis. It's in the spirit of a quantitative bias analysis to contextualize these inconsistencies and say, well, we know that there's probably relationships similar to this. From NHANES, we're going to apply these relationships and it's going to give us some way to interpret or sort of offer some contextualization for why we're getting this weird relationship between BMI and cancer mortality. And so I just want to acknowledge some of the limitations behind doing this. Is I used NHANES and I applied it to a study of head and neck cancer survivors. NHANES may not reflect these relationships among head and neck cancer survivors. So the relationship between BMI and smoking and so forth, that might look different in my population. So I'm sort of transporting that information into mine. And that's probably not completely legit, but I think that it's still informative in the sense of, in the spirit of a sensitivity analysis, we're sort of saying if this relationship holds, then this is what we would see. And I think that this is probably not that bad, that egregious of an error, because the questionnaire values for that study were done at the time of diagnosis, before treatment would probably affect their body composition or affect some of their other variables. So one of the key aspects that I think is really strong about this is that the Bayesian framework that I employed allows the uncertainty in the body composition prediction to propagate through to the hazard ratio estimates. So one of the things about the Bayesian paradigm is that you actually say that the thing you're imposing a prior on has a distribution associated with it. So there's error associated with things. So we didn't just predict body composition and act like we knew what it was. We said, okay, here's a model for body composition, and we're actually keeping the error term from our model in that prediction. And so this is something that can actually help address something that's ubiquitous in the application of prediction equations, which is called Bergson error. This is sort of roughly speaking when the error term that you have from a prediction equation, if you leave it out, you're basically sort of leaving some confounding on the table. The error term becomes correlated with your outcome, and you're not accounting for it. This allows us to at least account for it in at least a very cursory fashion. And there's been some discussions about the debates around this in the papers referenced below. But one of the ways that sort of advocated to at least somewhat address it is to just add some random error to your prediction that's built into the Bayesian paradigm. That's not actually something that's foreign to a Bayesian methodologist. So finally, I want to finish up with a quick talk about my thoughts about confounding in nutritional epidemiology. And confounding is a much broader topic than what I'm going to talk about today. But confounding in nutritional epidemiology, one of the sort of key features of confounding, I think, one of the key issues with confounding is that when we start thinking about individual foods or individual nutrients and their relationship with disease, we don't really take into account the fact that the other things that we eat might really be affected as well. So we tend to do things like put total energy in the model, or maybe adjust for vegetables or meat intake or something like that. But when we really think about comprehensive assessments of diet, we really should be thinking about all of the variables simultaneously. Diet is a multi sort of variable exposure. And so when we think about sort of the driving effects of diet, the nutrients or the constituents of our foods are really the active agents. We really think that these nutrients or these constituents are driving diet-disease relationships. And so the foods are actually what we're eating, and these things carry nutrients. And so, but it's likely other factors that we haven't identified. It carries nutrients that we have identified, but it probably carries nutrients or other things that we haven't identified. There's research going on every day to understand what things in foods are driving health outcomes. And so one of the things that I think is really interesting is that there's a lot of complex exposure patterns that are common in a lot of areas. So this happens in environmental epidemiology. We're exposed to a wide array of environmental chemicals. This is a factor in genetic epidemiology where we're affected by many genes or many polymorphisms. And so the method I'm going to present has actually been used a lot in both of these fields. And so it's been used a little bit in nutritional epidemiology, but I think that it could be considered a little bit more thoroughly. So one of the ways that I think a lot of folks have sort of proceeded with nutritional epidemiology is through what I would call serial analysis. And this is not analysis of cornflakes. This is analysis of single exposures at a time. And so you'll see a model that has meat in it, and then maybe some things that adjusts for kind of loosely. Then folks will run another model with another variable and another dietary variable like kale and so forth. And so you end up getting a lot of publications out of doing this, which sounds great, but basically it ignores confounding by other dietary exposures. And so when you see these, the fact that meat and kale intake might be inversely correlated is not actually accounted for in either one of these models. And so the models that sort of incorporate diet more holistically or more comprehensively are actually much better. But we still to this day have a lot of research that focuses on single foods or single nutrients. I think there's this temptation to sort of identify the magic pill in nutritional epidemiology. And I think that this is really a limiting issue in our field. And so I think this is really what's contributed to a lot of inconsistency in the epidemiologic literature. So I think a lot of folks are familiar with this paper of is everything we eat associated with cancer? And the authors basically randomly picked ingredients out of The Joy of Cooking, a very popular cookbook, and they searched the literature for studies that related each ingredient with different cancer outcomes, and you see these are the relationships here. You could maybe, if you squint hard enough, you can maybe say that there's some consistency here, but really there's a lot of spread around a lot of these. And so these are relative risk estimates. So you see relative risk estimates for something like bacon that are between probably 1.5 to 10. I mean, I think relative risk of bacon at 10 for cancers is absurd. And you see a lot of stuff that's in this really high range that I think is really unwarranted. Similarly, you see a lot of stuff in the low range that is also, I think, unreasonable. So I think nutritional researchers have some appreciation for this. And so I think this is driven a couple of approaches to doing this. The first is simultaneous exposure modeling. So this says we're going to take all the variables that we have on diet, maybe with some sort of aggregation, and we're going to throw them all together into one big model. Well, anybody that's done modeling will tell you that the more variables you do on the model, when you get 20 to 60 variables, you have a lot of problems there. And so this manifests with either wide confidence intervals or your model just doesn't run altogether. So in nutritional epidemiology, I think the dominant paradigm to try and tame this problem has been empirically driven data reduction methods. So things like factor analysis, principal components analysis, cluster analysis or latent class analysis tend to be really popular, reduced rank regression. So these are things that basically take the sort of correlation or the relationships between the variables and try to sort of cull them down to what combinations kind of travel together, either positively or negatively. And so I would say that, like, for broad strokes, this is not a horrible idea, but I think it really limits the interpretability, because now we've lost the ability to say that what does an increase in meat intake actually do, or what is an increase in leafy green vegetables actually associated with? So I think that this sort of abstracts things to the level that they might not be as useful or interpretable as we would like them to be. So a sort of application of Bayesian methods kind of emerged back in the mid 90s, where folks started realizing that we actually have expectations for how foods operate on health. They're nutrients. There's a lot of research around this. And so the idea became that similar nutrient compositions should be related to similar effects on disease risk. This is the driving factor of why we study nutrients, I think, in relation to disease outcomes. So the concept was we should shrink estimates of similar exposures closer together. So nutrients, the nutrient composition of cabbage is similar to broccoli, which is similar to cauliflower. They're not the same. We can accommodate differences, but there's a lot in common there that we can leverage to help sort of reduce the dimensionality of the problem. And so the concept of applying hierarchical models was introduced by Sandra Greenland back in 1993, and really advocated in the areas of genetic epidemiology, environmental epidemiology, and nutritional epidemiology. It really took hold in environmental epidemiology, I think, but it's sort of somewhat in genetic epidemiology, but I haven't seen a lot of applications in nutrition. I think it's actually really useful there. So just to kind of like give us a really quick introduction, I'm trying to keep the talk less mathy, but I can't help myself. So the model proceeds in two stages. The first stage is the model we typically would run. So we can kind of consider X being a collection of foods. It's a vector of foods that we want to model the effect of. Beta are the things that we actually care about. And we have another set of confounders that we're not going to worry so much about in terms of imposing some structure on them. You could impose some structure on those coefficients. The coefficient we care about is this beta coefficient. This is the effect of the foods on our outcomes. So what we're going to do now is we're going to model the coefficients on the foods, on our primary exposures, as a function of their nutrients. So we're basically saying that the betas, the coefficients from the first model, the coefficients that relate the foods to our outcome, are functions of the nutrients or some composition of those foods. That's what the second stage model is doing. So basically we have this other model, and then this delta, this sort of residual term, this is like a residual in a linear regression. This specifies the differences in the food. So foods with two similar nutrient compositions are going to have similar effects, but the deltas allow there to be some variability around foods with similar nutrients. So this allows so even if broccoli, cabbage, and cauliflower all had exactly the same nutrients, which they don't, but let's say they did, this delta will allow there to be some variability in the effect of each one of those on the outcome. And so this is a sort of very satisfying, in my mind, sort of approach to analyzing nutritional data, or dietary data. And so one of the interesting things, and some of the early applications of this did focus on foods as the primary components, but some of the later applications are actually focusing on considering the nutrients as our exposures. And I find that interesting because the nutrients are sort of a little bit closer to some of the biological mechanisms that we were thinking about. So in the application that my first application of this was looking at the relationship between different nutrients and breast cancer risk. And so we studied the intake of 33 nutrients for subjects. We scaled them to single standard deviation units. And then we created this thing, the second stage model that explains how the nutrients relate to the outcome based on the activity of the nutrients in different biological pathways. Admittedly, this is somewhat of a crude approach, but this is something that's been applied in other settings as well. Nutrients and neural tube defects by Susan Carmichael and John Whitty down at Stanford. They had done very similar analysis. Actually, this was modeled after that analysis. But we wanted to understand how these nutrients were related to breast cancer risk. So we're saying something like, So zinc, for example, just to pick one, is influenced by both, influences both the 1-carbon metabolism pathway, oxidative stress pathway, and glycemic control pathway. We did a comprehensive literature review. I had a great grad student that I was working with at the time that helped out with some of the biological mechanisms that I wasn't familiar with. But even though it's operating these pathways, it has its own effect that's independent of these pathways, that delta, that little residual variability. That's sort of the zinc specific effect that's not accounted for by influences in these pathways. So this actually kind of gives this really nice approach of integrating the biology into the analysis in a, I think, very thoughtful way. So these are these first stage estimates. These are the betas from that first stage model. And again, you don't see a lot of shift necessarily in the point estimates, but looking at the model with the single nutrient. So this is models that had one nutrient at a time. So a model with zinc, a model with carbohydrates, a model with each one of the 33 that we were talking about. The multivariable model throws them all in the model together. And the hierarchical model throws them all together, but then models them through this other stage. And again, one of the sort of striking things here is that you don't get a lot of movement in the point estimates, but you get better precision. It's because we're sort of reducing the dimensionality of the problem by doing this in a hierarchical setting. And so I think this is a really interesting way of approaching dietary analysis. Again, we're not getting tremendous insights, but I feel like we're getting more reasonable ranges on our credible intervals for something like this. One of the interesting things is you can actually interpret that the pieces of that second stage model as well. So the pi, the term pi here, which is the coefficients on the biological pathway covariates, you can interpret the pieces of that as well. And so the odds ratios that are associated with pi basically say that for a 1 standard deviation increase in the intake of a nutrient related to that particular pathway, this is the change in the relative odds of that. And so what was kind of interesting is we didn't really expect to see this, but the 1-carbon pathway, the oxidative stress pathway, and the phytoestrogen pathway tended to have really null effects. Again, these all include, all the credible intervals include the null, but the strongest signal that we had was glycemic control pathway. And this actually agrees with another analysis that I did in this population previously. And so this is just sort of really interesting, I think, that that we see that the glycemic control could be an important factor in breast cancer risk. And so I think that this also allows sort of again a more sort of comprehensive assessment of the relationships here. So instead of just looking at these results and then in the discussion sort of kind of like walking around and sort of like speculating that this could be driven by this pathway or that pathway or whatever, now we actually have another model that's built into our analysis that actually allows us to talk about the relative biological relationships. So I just wanted to highlight really quickly some recent work in this by my graduate student, Isaac Urgas, who just graduated this past summer. He applied this method to the study of diet and breast cancer survival in the pathway study. Larry Kushi down at Kaiser DOR is the PI. This is a prospective cohort of breast cancer survivors. And our goal was actually, we're kind of going back to looking at the sort of slew of foods that people consume. So we had a model that we wanted to analyze 28 food groups. We sort of aggregated foods a little bit into like broad groups, like meats, like green vegetables, and so forth. And we wanted to be able to model that as a function of the constituents of the foods, which are nutrients and other factors. And so again, no major insights. I think there's just a lot of variability in nutritional epidemiology that's going to be kind of hard to tame, but I think that we get like really interesting sort of more reasonable results here. We see this, so if you throw everything into one big model, we have this really absurd relationship with butter that was actually consistent when we did individual models. This actually looks like butter is protective for mortality among breast cancer survivors. It's a very wide confidence interval. But that's really, if you're going to interpret this literally, that's what what you're going to conclude. But in the hierarchical model that incorporated other aspects, the nutrients that are contained with butter, as well as other factors, then we actually see a more attenuated relationship with a tighter confidence interval that actually now includes the null. And so I think this is something that's kind of just really interesting in how we can sort of, when you think back to that paper by Schoenfeld and Ioannidis, you see like so many things that look really deleterious, or that look both deleterious and protective. I think this can sort of help address some of that, in the sense that we're kind of getting more precise estimates that are actually more reasonable when we incorporate multiple dimensions in the analysis. So in the interest of time, I see we're kind of getting low there, so I'll kind of skip that last slide. But Isaac's paper should be submitted soon, so hopefully you can read about that then. So I just kind of wanted to highlight this methodology that I've just applied is not, or just described as not unusual. It's been applied a lot in environmental epidemiology, somewhat in genetic epidemiology. And I think that environmental epi and nutrition epi share a lot of these same issues with regards to multiple exposures. And I was actually invited to talk on this topic at a symposium at SER, where we discussed this and sort of the different ways the different fields are addressing this. And environmental epi really likes this approach. And I think that this could be applied a lot in nutritional epidemiology as well. And so one of the things I really like about this is that it encourages you to engage with your subject matter in a way that I think in nutritional epidemiology we don't always do. We maintain the inference on their unit of exposure, and we have better precision, which I think is really one of the key factors for this. So some final thoughts about this is the purpose of this talk is not to convert folks to be Bayesians, but to sort of highlight how this paradigm can be useful to address some of these issues in nutritional epidemiology, but also more broadly in other fields of epidemiology too. And so hopefully I've sort of at least kind of sort of highlighted some of the things to kind of make you aware of some of the ways that this can be used maybe in your research or sort of just to help contextualize some of the other findings. And so kind of keep in mind a couple of things. Bayesian models, there's nothing inherently useful or key about these things being Bayesian. We can oftentimes specify what we're doing in the Bayesian framework in a frequentist framework. It's more that it's a bit of a philosophical bent about like what these things mean, but also that it's actually just easier to do these things a lot of times in Bayesian frameworks. And so in terms of being able to, instead of maximizing a complicated likelihood, which can be problematic, we can characterize the posterior distribution sometimes much more straightforwardly. But one thing to caution is that we use Markov chain Monte Carlo or these sort of random sampling tools to characterize the posteriors, but you can actually specify overly complex models pretty easily in the Bayesian framework as well. So something to kind of keep in mind there. I want to acknowledge my collaborators and some things that supported the funding of the studies that I just mentioned. And with that, I will end here and I will take questions.
Great, thank you so much, Patrick. Questions? People want to pipe in or add a question in the chat? Hi, Steph, do you have a question?
I don't. I was just going to say thanks. Interesting. I have to process it to think about how it might be applicable in my work.
Thanks. Yeah, that was a little bit of a drinking from the fire hose kind of talk. I apologize if it was. I felt a little like I was doing a brain dump a little bit, but yeah, there's a lot of applications. I mean, I think that there's some econometric applications too of Bayesian methods that I think are very tightly tied to some of the things that I've talked about. The sort of selection models, like the Heckman models, are sort of related to the selection model that I was talking about for survival analysis. The problem is like in survival analysis, we can't actually like specify a single joint distribution the way we can like in a Heckman model, for example. And so we do that sort of cruder partitioning, I think. But yeah, there's a lot of applications to these methods. I think that would be really useful broadly.
Great. Well, thank you so much. Really appreciate the talk and a lot of food for thought. And I'm sure people can reach out to you with more questions as they think about it. But thank you so much. Really,
absolutely.
Really appreciate it.
Thanks for having me.
All right. Thanks, everyone. Bye-bye.